The overall goal of this chromatin immunoprecipitation protocol is to determine the abundance and localization of histone modifications at defined regions throughout the budding yeast genome.
This method can help answer key questions in chromatin regulation, such as how histone modifications are linked to gene expression control and how these modifications change in response to different environmental conditions.
This is an extremely versatile protocol that can be used to investigate many different histone modifications in multiple yeast strains at one time.
Though this method can provide insight into histone modifications, it can also be applied to investigating other chromatin-binding proteins through the use of epitope tags or available antibodies.
Start this procedure by growing the yeast cells. Inoculate 10 milliliters of YPD medium with a single colony of each appropriate strain, and grow at 30 degrees Celsius in a shaking incubator at 220 RPM overnight. On the following day, after measuring the optical density at 600 nanometers or OD600 of the culture, dilute the culture to an OD600 of 0.2 in 100 milliliters of YPD in a new flask. Grow the cells at 30 degrees Celsius in a shaking incubator at 220 RPM until they reach mid-log phase. When the cultures have reached mid-log phase, add 2.7 milliliters of 37% formaldehyde directly to the medium in each flask for a final concentration of 1% formaldehyde. Transfer the flasks to a shaker at room temperature and shake at 50 RPM for 15 minutes. Add 5 milliliters of 2.5 molar glycine to the medium and continue shaking at